Glycine max. Miracle beans, golden beans, king of beans. Soybean goes by many names. It's a powerhouse crop, producing protein for human food, animal feed, edible oils, and even biofuels. Its ability to work together with microbes to acquire nutrients reduces the need for fertilizers, making it an ideal crop for sustainable agriculture. Today, soybean is one of the most consumed crops in the world. In the 2024 season alone, global production reached 394 million tons. That's like stacking over 39,000 Eiffel Towers on top of each other. But there's a catch. Around 80% of global soy production comes from just 3 countries: the United States, Brazil, and Argentina, leaving market trades heavily reliant on imports. This reliance means a bigger carbon footprint from long-distance shipping and increased deforestation to meet growing crop demand, making soybean costly for the environment. But what if we could grow soybean closer to home? That's exactly what the French company Sangosse and the Sustainable Project are working on, expanding the cultivation of soybeans to northern France. Sounds simple? Not quite. Northern France isn't Brazil. It's colder, and soybeans don't like the cold, especially when they're young. And climate change makes this even more complicated. With temperature shifts and unpredictable precipitation, farmers face a gamble. Plant too early, and a sudden cold stress could stunt the seedlings. Wait too long, and the crops risk waterlogging when maturing. One promising solution lies beneath the soil, literally, and here's where our research comes in. Plants have tiny allies hidden in the soil, their microbiota, a community of microorganisms including bacteria and fungi that can help plants grow. We are focusing on bacteria, which can thrive in diverse conditions, including the cold. Research shows that these organisms can help plants tolerate the cold by secreting biomolecules that support plant growth. Our project focuses on harnessing and manipulating soil bacteria to help soybean tolerate the cold. We aim to determine if bacterial strains tolerant to the cold can also induce cold tolerance in soybean plants by assembling synthetic microbial communities to observe their effect on plant growth. As a first step, we identify and genotype bacteria from soybean fields through visual characterization and sequencing. Then we test which of these bacteria can grow in cold temperature and assess some beneficial functions, such as making nutrients available to the plant or producing secondary metabolites to shield against diverse stress factors. From these results, we will be able to create several syncoms to inoculate soybean plants with and observe their effect on growth under cold stress. This will be determined by comparing treated and untreated plants based on physiological measurements of traits related to growth, as well as the expression of genes related to cold stress from RNA sequencing. With our project, we aim to study biological solutions to tackle the cold as a crucial limiting factor for soybean growth in Northern Europe. This will help pave the way for more sustainable soybean cultivation, with fewer imports, reduced carbon footprints, and more resilient crops, all thanks to the hidden power of soil bacteria.